In order to meet the demands of constantly changing fashion trends, retailers are producing cheap, low-quality clothing in the industry come to be known as fast fashion. Consumers are able to buy more clothing more frequently at a lower price point. But this convenience comes at a cost. The fast fashion industry results in the creation of 92 million tons of textile waste each year across the globe, uses 79 trillion liters of water, and accounts for 10% of human greenhouse gas emissions. And this industry is only growing. 60% of clothing on retail shelves today is made from polyester, an artificial fiber that can take up to 200 years to degrade naturally. Polyester has become so prevalent that in Canada alone, it accounts for 7% of all plastics in landfills, making it the third largest category of plastic waste in the country. While it may take a shift in societal values to alter the fashion industry, we can reduce the environmental impact of the industry by making informed choices and by disposing of our clothes responsibly. We are RHS-Calgary, and to combat this problem of fabric waste, we present our solution, FabriFuel, an innovative and refreshing approach designed to address fabric waste while promoting sustainability. We aim to address this problem of fabric waste by designing a microbial fuel cell capable of digesting polyester as its primary energy source. The project consists of two main objectives. We aim to 1. Reduce the environmental impact of polyester waste, and 2. Capture the energy produced as a byproduct of this digestion. It is our hope that the production of energy from these microbial fuel cells will incentivize adoption of these cells and make them more cost-effective. In this, our product promotes both environmental and economic sustainability by reducing harmful wastes in our environment and producing greener and cleaner energy. There is a current lack of cost-effective and sustainable ways to recycle polyester and other plastic-based textiles, which leads to waste and the creation of toxins. Recycling methods are also not promising, as recycling polyester requires many harmful chemicals, including chlorine, and results in the release of microplastics that can further harm the environment. Our project involves two main methods of innovation. Firstly, our wet lab team is working towards creating a genetic circuit to digest polyester in E. coli. Secondly, our dry lab team is working towards engineering the microbial fuel cell that will house our genetic system and capture the current produced by the E. coli through the metabolism of polyester. In the design of our project, we first began by conducting extensive research to find a naturally occurring bacteria that is able to digest PET, which is the abbreviated term for polyester. Such a bacteria is Ideonella sicyensis, which secretes two enzymes, which, together, are able to break down PET into its monomers. This two-enzyme system consists of metase and petase. Previous research done by Dr. G. Beckham in Colorado has shown success in expressing a chimeric protein that combines both petase and metase from a single plasmid. We intend to use this already established plasmid in our work. PET is digested by these enzymes, producing various compounds and byproducts, the key end product being acetate. Acetate can be oxidized and used as an energy source by E. coli, which will in turn generate an electric gradient in our microbial fuel cell. So far, we obtained the plasmid from Dr. Greg Beckham's lab through Agene and have been working on preliminary experiments. These include transforming the plasmid into DH5-alpha, our selected strain of E. coli, and producing DNA stock through mini-preps. We are currently working to verify the DNA yield and intend to complete a restriction analysis in the near future. The second part of our project is to utilize the energy released by the bacteria using a microbial fuel cell. The dry lab team is working to implement a microbial fuel cell that will allow us to capture the excess energy released by the bacteria. Microbial fuel cells work much like a battery. There are two chambers separated by a semi-permeable membrane, each containing a conductive surface. One of these surfaces acts as the anode and will be placed in the chamber with the bacteria, while the other will be the cathode and will be exposed to the atmosphere. The movement of electrons is a key part of a bacteria's metabolic process, and the microbial fuel cell seeks to take advantage of this by acting as an acceptor of electrons. The electrons that gather on the anode will flow to the cathode, resulting in an electric current. These are basic principles of all microbial fuel cells. The unique feature of our microbial fuel cell is that the bacterial population will be able to digest polyester. Once we have reached the stage of having a functional prototype, we will be testing our genetically modified bacteria in a variety of conditions to determine the efficacy of our system. These conditions include temperature, pH, different growth mediums, and different elements in the design of our microbial fuel cell. While the wet lab and dry lab portions of our team have been working closely together to bring this project to fruition, our human practices team has been working to contact experts in our community and to raise awareness of this issue. Particularly, our human practices team has worked with a few key individuals who have aided us in this project. Firstly, Robert Mayle and Emily Hicks, co-founders of FredSense, have been our leading mentors who provided us with information on the fundamentals of our project, helped us predict and circumvent potential roadblocks in our prototype, and gave us unconditional support on how we could best develop our idea. Secondly, the research from Dr. Beckham's lab gave us the means to begin our research. Thirdly, Mr. Jason Brannick from Canadian Natural Resources Limited has provided us with guidance on the commercial reliability of this project. Finally, a great depth of gratitude goes to our teachers, Dr. Iyasi Suarez, Ms. Kim O'Keefe, Ms. Dallas McLaughlin, and Ms. Alyssa Crispin for providing direct guidance and support as they have guided us throughout this project. Overall, we are grateful for the opportunity to be able to work on this project, especially since the issue of fabric waste can often be overlooked. We, as consumers, enjoy choice in what we wear, but oftentimes we don't see the immense negative effects of these choices. Fast fashion, a rapidly growing industry in our world, has been recklessly encouraging wasteful consumer choices. With its growing prevalence, there comes a wide array of environmental issues stemming from fabric waste, which, if not addressed now, will only continue to drastically affect the world we live in through increased greenhouse gas production, excessive water depletion, and much more. It's important, now more than ever, that we take action and begin to solve the problem of fabric waste. We are RHS-Calgary, and we created FabriFuel, an innovative approach to solving this problem of fabric waste. FabriFuel, reducing waste, producing energy, all in a single solution.